Welcome to your channel Vegyanghar. In this video, we are going to talk about IIT JAM 2020 Biotechnology paper, which was organized by IIT Kanpur. This paper is really important because it gives you an opportunity to get admission in top institutions across India, including IITs and various IISers. This paper comprises of three sections: Section A, Section B, and Section C. In this video, we are going to talk about Section A. The Section A contains a total of 30 MCQ based questions. Each question the first 10 question carries 1 mark and the left 20 question carries 2 marks each. For every wrong answer, for all 1 mark questions, 1 by 3 marks will be deducted, and for all 2 marks question, 2 by 3 marks will be deducted for each wrong answer. This section comprises of biology, physics, chemistry, and mathematics. All you need to know is that this section carries going to ask you questions which are very basic, factual, to the point. So let's start. The first question is, deficiency of enzyme phenylalanine hydroxylase causes phenylketonuria. Phenylalanine hydroxylase converts phenylalanine to tryptophan, alanine, tyrosine या threonine. So now we know that जो phenylketonuria है, it is an autosomal recessive disorder and it is caused because of the deficiency or non-functional of this enzyme hydroxylase. What is the function of this hydroxylase enzyme? Is that it attaches OH group to the phenylalanine amino acid, which results in a new amino acid called as tyrosine. So yes, option C is my correct option. The next question is, T cells and B cells are. Now I am very sure that you know already the answer, that is lymphocytes, right? But let's look into a little detail. Now this is a picture taken from the book Kuby, and you can see that in the initial developmental stages of our life, we have a hematopoietic stem cells. I mean those cells which are going to give rise to the blood part, right? Now hematopoietic stem cells can be ultimately divided into two. We have myeloid progenitor cells and we have lymphoid progenitor cells. The lymphoid progenitor cells are going to give me our asked cells, that are T cells and the B cells, and they are commonly called as lymphocyte. Now why they are calling as lymphocyte? Because they arise from lymphoid progenitor cells. Apart from this lymphoid progenitor cell, we have myeloid progenitor cell also. In one of the paper they asked myeloid lineage, so one has to know also that myeloid progenitor cell give rise to dendritic cell. Apart from dendritic cell, we have granulocyte like monocyte, eosinophils, basophils, and megakaryotes, which ultimately give me platelets, and ultimately yes, erythrocytes also. So yes, for this question the answer is option A, lymphocyte. The next question is, in humans, the testis temperature is maintained below the core body temperature with the help of. We know that testes in males are responsible for a process called as spermatogenesis. This spermatogenesis process is responsible for production of sperm. For this production, the testes require a temperature below than the body temperature. यानी कि नॉर्मल बॉडी टेम्परेचर दैट इज 98.6 डिग्री फ़ारेनहाइट या 37 डिग्री सेल्सियस से कम टेम्परेचर, कितना कम, 3 से 4 डिग्री सेल्सियस कम चाहिए हमारे टेस्टिस को. एंड देयरफॉर दिस रिक्वायरमेंट इज सेटिस्फाइड with the role of scrotum. Now scrotum is a pouch like structure which contains testis and give a particular environment which contains a temperature lower than the normal body temperature. So yes, option C is my correct option. The next question is, the correct sequence of phases during mitosis is. We know that mitosis is an equational division. The mitosis comprises of two portions. We have the M phase and we have the interphase. In interphase we have G1 phase, S phase and G2 phase, while in particular mitotic phase, that is the M phase, we have four major phases, that are prophase, metaphase, anaphase and telophase. So yes, the correct sequence is prophase, metaphase, anaphase and telophase. Before moving on, in prophase, the chromosomes are being formed, that is the condensation of chromatin which results in chromosome takes place. Then we have the metaphase. In metaphase, all the chromosomes are going to be aligned in a particular equatorial plate. In the anaphase, we have a complete separation of sister chromatids, and in telophase this process completely takes place, and after which cytokinins begin. So the option A is correct, that is prophase, metaphase, anaphase and telophase. Next question is, protein that help other proteins to fold correctly is. Now we know that protein folding is not a random process, but it is a guided one, right? Now in this folding, we have some other protein which are necessary, like chaperones. Now chaperones are those protein which are responsible for protein folding. But let's see what others options are responsible for. For proteasome and ubiquitin, both are responsible for protein degradation, while desmosome is going to function as a intercellular junction. You also need to note down that desmosome is a modification of Golgi apparatus, right? So yes, chaperones are those protein which help other proteins to fold correctly. That is option A is a correct option. Next question is, in viruses, capsids are made up of. Now we know that the outer covering of virus is called as capsid, which is made up of monomer called as capsomeres, right? These capsomeres are protein by nature. So yes, capsid are made up of protein. Option A is a correct option. The next question is, the fungal cell wall and the crustacean exoskeleton contains a common component. What is that component? Now that component is basically chitin. We know that chitin is a biopolymer of NAG, and it is one of the main component of, as I said, cell wall of fungi, exoskeleton of arthropods and crustaceans, insects, the radule of the mollusk and the beaks of cephalopods. Now apart from chitin role in fungal and cell wall, let's look into cellulose. We know the plant cell wall is made up of cellulose. Peptidoglycan is present in the cell wall of bacteria, while lignin is the second most abundant complex polymer, which is a key structural material in the support of tissue of vascular plants and some algae. So yes, for this question, the common component in crustaceans exoskeleton and fungal cell wall is chitin. Option C is correct answer. Next is, the correct sequence of evolution symbol to complex is. Now we know that plants are of different types. On the basis of evolution, it was the algae which arose first, then we have bryophytes, then we have pteridophytes, gymnosperm and then angiosperm. Fern belong to pteridophyte. So yes, option A is correct. That is algae, bryophytes, fern and angiosperms. Next question is, during El Nino. Now El Nino is basically a weather phenomena which takes place because of cold water of north flowing Peru current displayed by the low nutrient warm southern current. Now one has to understand this is a factual question and one has to cram the answer. That is option A is the correct answer. So please note the important terms, which are low nutrient warm southward current and cold water of the north flowing Peru current. So yes, option A is a correct option. Now the next question is a match the following question, where group 1 carries the deficiency diseases and group 2 carries deficiency diseases associated vitamins. So we know that various deficiency diseases are caused by deficiency of various vitamins. In this question, let's first of all, you have to mark what you know. I know that night blindness is caused by deficiency of vitamin A, but in group 2 you cannot able to find vitamin A. It means that one has to know the chemical name of vitamins. So vitamin A is also called as retinol, vitamin B complex is there, vitamin C is ascorbic acid, vitamin D is calciferol, vitamin E is tocopherol and vitamin K is phylloquinone. So let's see the answer. We know that night blindness is caused by vitamin A, which belongs to chemical retinol. Then we have beriberi. Now we know that beriberi is basically caused by deficiency of vitamin. Yes, so we know that beriberi is caused by thiamine deficiency, vitamin B1. Then we have scurvy. Scurvy is caused by deficiency of vitamin C, ascorbic acid, and birth defect are related to various folic acid. Now folic acid is not a direct vitamin but a derivative of vitamin. So option C is a correct answer. Next question is eutrophication refers to an aging process from a. Now we know that eutrophication is an aging process. Now what is eutrophication? It is when a body becomes overly enriched with minerals and nutrient which can induce excessive growth of algae. Now because of this enriched nutrient, the production of ecosystem which was initially at low becomes high. Okay, so yes, option A satisfies our need, that is low production ecosystem to high production ecosystem due to availability of excess nutrients. Okay, so next question is match the enzymes in group 1 with the corresponding substrate. Now in group 1 we have different enzymes and then we have substrate. This is a factual question, you have to understand this thing. Amylase is responsible for starch degradation. Pepsin is responsible for protein degradation. Lipase is associated with fat metabolism, while ribozyme is responsible for RNA degradation. So yes, option B matches the correct options. So yes, option B is a correct answer. Next question. In ABO blood group testing, which one of the following is incorrect? Now, one has to understand that, during blood grouping, the blood donor and the blood recipient should match each other with the following. It means that if I'm going to, transfer blood group B to a person with blood group A, the person with blood group A is going to, take blood group B as a foreign antigen and is going to react. It will result in various biotransmission reactions. Therefore, it is very unhealthy. Here, option C is correct. Now, why option C is correct? Because AB blood group contains both the antigen of A and B. It means that it will have agglutination. It means it will show reaction with anti-A or anti-B bodies. And that is why this option is incorrect. It is showing no agglutination, but yes, there will be agglutinization. Now, what is agglutination? जब भी कोई antibody किसी structure react करता है, it will lead to precipitation, and this complete phenomena is called as agglutinization. So yes, here option C is incorrect. I mean, option C is correct, and according to question, it is incorrect, right? Next question is match the RNAs with their corresponding functions. Remember one thing, that all RNAs are not coding RNAs. There are something called as non-coding RNAs which are responsible for various cellular processes. Some of them mentioned are there. We know that snRNA is responsible for RNA splicing, while the piRNA is responsible for germline protection from transposable elements. Transposable elements are also known as jumping genes. Then we have snRNA. snRNA is responsible for rRNA processing, while the siRNA is responsible for selected mRNA degradation. It should be noted down and should be remembered. Okay. Next question. What match the type of bacterial flagella? Now this question is again a match the following question, where you have to know that a bacteria for its locomotion uses flagella. This flagella can be of varies in number. We have some bacteria which contains only one flagella at one polar end called as monotrichous. If a bacteria is containing more than one flagellum but only at an end, only at a single end, it is called as lophotrichous. In peritrichous, when the bacterial cell surface is covered with many flagella, and in amphitrichous we have flagella at two polar ends. So according to our need, option B is found to be the correct option. Now next question is based on assertion reasoning. Assertion में आपको एक statement दिया रहेगा and reason में assertion का explanation. Okay, let's see. The difference in the respective melting point of butter and coconut oil is caused by degree of saturation of the corresponding fat chain. Assertion ये कहना चाहता है कि butter और coconut oil में जो fatty acid chains होते हैं, उनका जो boiling point है, okay, I mean जो उनका boiling point है butter और कोकोनट ऑयल का, वो डिपेंड करता है कि उसके अंदर जो फैटी एसिड चेन्स हैं वो कितने डिग्री पे सैचुरेटेड है. नाउ दिस असर्शन इज करेक्ट. ओके, जितना ज्यादा सैचुरेशन उतना ज्यादा मेल्टिंग पॉइंट, जितना कम सैचुरेशन उतना ज्यादा फ्री. नाउ रीजन इट सेज unsaturated fatty acid chain in fats and lipid becomes solid more easily because they are relatively straight and thus able to pack together more closely than saturated chain. Now this is wrong. Why? Because unsaturated fatty acid chain shows a kink, it shows a bend which result in unfitness, यानी कि वो एक साथ काफी मतलब काफी अच्छे मैनेजिंग स्टाइल में स्टिक नहीं कर पाता. It means it is not going to be closely packed. Therefore, the reason is holds false. It means the option D is correct. A is true, that is assertion is true, while reason is false. Now again assertion reasoning. We know lac operon is an inducible operon. It means that this operon can be induced to on state by giving lactose. So assertion is correct. Now, lac operon is not induced when the repressor protein remains bound to operator DNA sequence. It means that in the absence of lactose, जो हमारा repressor protein है, वो operator sequence पे bind रहता है, जिसके कारण proper translation हो नहीं पाता, transcription हो नहीं पाता, and therefore lac operon is switched off. It means yes, assertion A and reason R both are correct, but reason is not giving me an absolute explanation of assertion. So yes, option B is going to be my correct option. Next question is match the technique in group 1 with their application in group 2 for protein analysis. Now we know that we have various technique to understand various protein, its proximity sites, its concentration, its size, its structure. उनमें से कुछ यहाँ पर आपको मेंशन की गई हैं. NMR, that is the nuclear magnetic resonance spectroscopy, is one of the most widely used spectroscopic method to analyze the structure of a protein. So P4 is going to be my answer. And now you can see that P4 is an exclusive to option B. Option B is going to be my correct answer. The rest belongs to fluorescence resonance energy transfer is used to find the proximity of two sites located in the protein. दो साइट एक प्रोटीन में किस तरह से कितनी दूरी पे अटैच है, क्या रिलेशन है उनके बीच में, उसको स्टडी करने के लिए होता है. While the UV spectroscopy, that is the ultraviolet spectroscopy का काम है प्रोटीन के concentration को पता करना, while the dynamic light scattering is used to understand the size of the protein. So option B is the correct option. The next question is, the spectrum of a compound C5H10O shows a band at 1750 centimeter inverse. The same compound showed two signals, a triplet and a quartet, in its NMR spectrum. Identify the compound from the following. Now, this is one of the favorite questions asked by IITs in this paper. So here we are going to discuss about two different techniques which is used to study different compounds associated with protein or protein itself. Now I recommend all the viewers to make some points. One of the point I would like to mention is that when the IR reading shows 1700 plus centimeter inverse, it means that the compound is going to have a carbonyl oxygen. It should be clear. It means that in the question it is given that 15 centimeter inverse, I mean 1715 centimeter inverse is a mark that the compound is carrying carbonyl group. Now one thing is clear. Now if I'm going to look at the options, all of the options is going to have carbonyl. The A, B, C, D, all of them is having carbonyl. Cool. So okay. So the next, the next clue which is given in this question is that the compound is showing two signals, not only not one, not three, but distinct two signals, a triplet and a quartet. So first of all, looking at the options A, B, C and D, you can see that in option A, C and D, all of them is going to give me multiple signals because the hydrogen is going to react differently as they are very distinct from oxygen. There is lack of symmetry. But interestingly, in option B, we have a symmetry due to which there will be a two distinct signal and not multiple signal, but two distinct signal. Now to verify it, let's look at the NMR spectrum after the nuclear over-Haizer effect. Now nuclear over-Haizer effect हमें help करता है ये बताने के लिए कि हमारे आसपास कितने हाइड्रोजन है. So in this picture, you can see that the compound B is written. Now, if I'm going to look at the A portion, the terminal A position, you can see that the methyl group is having, methyl group के पास दो हाइड्रोजन है आपके पास, right? Now this two hydrogen which is attached directly to the carbonyl group is going to give me triplet signal. इसके पास दो हाइड्रोजन है और न्यूक्लियर ओवरहाइजर इफेक्ट के बाद ये जो दो हाइड्रोजन है उसमें आप 1 प्लस कर दीजिए तो यू विल गेट ट्रिपलेट सिग्नल. फाइन. उसी तरह से CH2 के लिए CH3 के हाइड्रोजन इज इंपॉर्टेंट. ओके, सो CH2 के सिग्नल्स के लिए CH3 अगर हम देखें तो CH3 के पास 3 हाइड्रोजन है, right? And according to nuclear overhizer effect, NOE effect, you will get CH3, that is 3 hydrogen plus 1, that is 4. It means that हर CH3 के लिए CH2 उसके पास में है and that is why we are going to get a triplet signal, that is 2 plus 1. और हर CH2 के लिए उसके नजदीक 3 हाइड्रोजन एटम्स है, right? So 3 हाइड्रोजन एटम्स है तो 3 plus 1, that is quartet. That is why this compound is the most suitable compound for this question, and hence option B is a correct option. Now, so this the complete अभी हमने जो पिछले questions किए वो सारे bio-related questions थे. Now let's move on to little maths, little physics, and yes, my favorite chemistry. Let's see. The curve y is equal to x to the power 4 minus 4 to the power x cube plus 4 to the power x square minus 4 has a tangent parallel to the x-axis at the following points x and y. Now you have to find the coordinates of that, that tangent which is passing to this curve. For that, one should know that the slope of the curve is given by dv by dx. It means that you have to differentiate the equation with respect to x. After differentiation with respect to x, you have to equate the opposite with respect to 0. And ultimately, you are going to find the coordinate of x. That will be either 0, 1, and 2. So, look at the coordinates of x-axis. So, option B matches with our system. So, option B is a correct answer. The next question is, for an ideal gas at room temperature, choose the correct representation of the Boyle's law. Now, first of all, you have to understand what is Boyle's law. So, Boyle's law states that the pressure of a given quantity of a gas varies inversely with its volume at a constant temperature. Remember, आपको ये याद रखना बहुत जरूरी है कि ये जो law है वो क्या कहता है? ये कहता है कि कांस्टेंट टेम्परेचर पे जब हमने किसी सिस्टम का टेम्परेचर कांस्टेंट कर दिया तो उसके अंदर का जो प्रेशर और वॉल्यूम है वो दोनों इनवर्स प्रोपोर्शनल में अपने आप से रिएक्ट करेंगे. इसका मतलब ये कि जब हम प्रेशर को इंक्रीज करेंगे द वॉल्यूम विल डिक्रीज, और जब हम वॉल्यूम को हाई लेवल पे मेंटेन रखेंगे तो उस पूरे सिस्टम का जो प्रेशर होगा वो बहुत लो रहेगा. फाइन. नाउ लुकिंग एट द ग्राफ थर्ड, P and 1 upon V, you can see that it is a straight line, right? You also have to understand that the x-axis and y-axis should be written, correctly. Here, option 3, graph 3 is a correct option because we can see that pressure is directly proportional to 1 by V. Remember, directly proportional to 1 by V. But yes, it is inversely proportional to V. Fine. So option C, that is a correct option. The next question is, if a coin is tossed 3 times, what is the probability that no 2 successive tosses shows the same face? इसका मतलब ये है कि अगर मैं किसी कॉइन को 3 बार टॉस करूं तो 3 बार मुझे 3 अलग अलग आउटकम्स मिल सकते हैं, राइट? मुझसे पूछा गया है कि कितने ऐसे इवेंट्स होंगे जिसपे जो कॉइन का फेस है वो सेम ना आए. Okay, so let's look into it. So first of all, let's find out how many possible events are going to be there. So possible combination for such event is 2 to the power cube, that is 8. Now these are the possible situation you can get, out of which 2 of them shows no 2 successive tosses, like head tail head and tail head tail. Okay, so therefore the probability is 2 by 8, that is 0.25. Option A is my correct option. Next question is, in a typical mammalian cell, the protein content is 20% of its net weight. If the density and the volume of the cells are 1.2 gram per ml and 4 into 10 to the power minus 9 ml respectively, then the concentration of the protein is. Now you should note that density is mass upon volume. So mass is going to be density multiplied by volume. After calculating the mass, we know that 20% of the protein is of mass. So took out the 20% of that. So we get 0.96 into 10 to the power minus 9. After that, we know that concentration is going to be net protein content upon cell volume. Cell volume is given as 4 into 10 to the power minus 9 ml. We have calculated, it comes out to be 0.24 gram per ml, right? But we have to convert it into milligram per ml. So we multiply by 1000 basically, and you ultimately यहाँ पर 100 नहीं आएगा, यहाँ पर 1000 आएगा. 1000 को multiply करेंगे तो we will get 240 milligram per ml. It means option D is my correct option. The next question is, consider a spherical particle of mass m and radius r moving in a medium. Now its velocity at time t is given by v is equal to v naught exponential minus 6 pi x rt upon m, where v0 is an initial velocity of particle, the dimension of x will be. Now to find the dimensions, one has to remember one of the most simple concept of class 11, that is principle of homogeneity. Now according to principle of homogeneity, same units can be multiplied or divided or plus or subtracted, right? Now according to following it, let's put up, we know that m is in denominator, the mass is in denominator. It means that the above unit should be mass. So while comparing the numerator with the denominator dimensions, we get xlt is equal to m, and hence x is going to be m l minus t minus 1, right? So option C is my correct option. Moving on, the next question is, consider a monoatomic ideal gas molecule of equal mass m in thermal equilibrium at temperature T. Which one of the following equation is correct? Now we know that monatomic ideal gas से मतलब है हमारा कि एक ऐसा गैस जो मोनोएटॉमिक हो, यानी कि सिंगल एटम से बना हो, इसका मास m हो और अभी वो थर्मल इक्विलिब्रियम में है. मुझे पता है कि कोई भी मॉलिक्यूल का जो अगर थर्मल इक्विलिब्रियम में है तो उसका जो काइनेटिक एनर्जी होती है वो होती है 3/2 kBT, which is very much equal to half mv square. Now this is a formula based question. You have to remember the formula, and option B is my correct answer. Fine, cool. Moving on, the next question is, the following carboxylic acid have a general formula R-COOH. Now R-COOH, okay, we have four options. Which of the following represent the decreasing order of the respective pKa values? Now for solving this question, one should note that higher the pKa, lower the Ka, hence lower the acidity. Lesser the, okay, so what I'm saying is that when a molecule is higher acidity, it is going to have a higher value of Ka and a lower value of pK. While vice versa, higher the pK, lower the Ka, and hence lower the acidity. Interestingly, in a molecule, lesser the withdrawing group attached, lower the acidity, and therefore higher the pK. The second line is very important, and consider all the options 1, 2, 3, 4 of carboxylic acid with this statement. What I'm saying is that in the option A, that is HCOOH, we have no methyl group. In option B, in second case, we have only single methyl group, while in third we have one chlorine also attached, and in fourth we have 3 fluorine attached. It means, okay, we know that electromagnetic elements are electron withdrawing in nature. It means they are withdrawing group. Okay, it means that option B is going to be my correct answer. That is, the carboxylic acid associated with the methyl group is going to be, have a higher pK and a lower acidity. Then we have the normal HCOOH, and then we have the carboxylic acid with a single Cl, and then finally at the end, the highest acidity and the lowest pK value is going to be CF3COOH. So option B is correct. Please note the principle. The next question is, which of the following compound is the simplest alkane that is optically active? Now, to be optically active, one should note that a compound should have, the one of the observation is that the compound should have, first of all, odd number of carbon atoms. Along with that, one has to also understand that it should, when the mirror images should not overlap. Fine, they should not overlap. Now in option A and C, the total number of carbon atoms are 7, while rest two have 8. So answer has to be from A and C. Okay, that should be clear. In option A, molecule has a chiral center. Remember this thing, and it also has a no chlorine. Hence, it is simplest. So please note the principle, and option A is my correct option. The next question is, in the following reaction, X is an intermediate and Y is one of the end products. Which one of the following compound is the end product Y? Now, this is an organic reaction associated with Grignard reagent, RMGBr. I hope you remember the class 7th concept of, class 11th and 12th organic chemistry. And following that concept, what we know is that when the associated reactant is going to react with our Grignard reagent, we will have this intermediate, which is shown as X, right? Now this intermediate is going to react with this carbonyl group, right? And after proteolysis, the carbonyl group will be, this IMGO group will be removed, and in the presence of H plus, it is going to be protonated and we'll get an OH, terminal OH group. It means that Y is my intermediate, and therefore option D is my correct option. The question says, which one of the following ion has the maximum number of unpaired electrons? Now we know that in a soft shell we have two electrons, right? A pair of electron, and if one electron is missing, it is termed as unpaired electron. Now we are given four cations. We have Cu2 plus, Na plus, Cr3 plus, and Fe3 plus. After writing its configuration, considering the loss of 2 plus, plus, 3 plus, and 3 plus electrons, we get an electronic configuration of Cu2 plus, Na plus, Cr3 plus, and Fe3 plus, according to Aufbau principle. Now you know that in D subshell, a total of 10 electrons can accommodate, and in a P subshell, a total of 6 electron can accommodate, right? So in case of Cu2 plus, you can see that the 3d9, it is almost filled and we are left with a single unpaired electron. 2p6 in sodium, P is completely filled. In chromium 3 plus, 3d2, it means that out of 10 electrons, we have only 2 electrons which are unpaired. Okay. Interestingly, in case of Fe3 plus, the 3d5, it is half filled configuration. And remember, half filled configuration is an unpaired configuration also. It means that Fe3 plus 3d5 configuration is going to give me the maximum number of unpaired electron, and hence option D is a correct option. I hope guys you like the video, and I'm sure you're going to score well. Thanking you. Like, share and subscribe. Thank you for watching vegyanghar.